Good afternoon, everyone. Thank you, Dr. Mano Maurya, for the introduction. Today I'm going to talk about how at Metabolomics Workbench, our National Metabolomics Data Repository, we are facilitating sharing, analysis, and integration of metabolomics data. So this work is really a culmination of effort over more than 20 years by a number of people and funding from various resources, mainly different institutes of NIH. And I'm presenting this work on behalf of the NMDR team, of which Professor Subramanian, who is the PI for this grant, and Dr. Owen Fahey, who is really the core person to make this resource available. So let's see, where do the metabolites fit in the cellular context? We have genes, proteins, enzymes, metabolites. So metabolites are essentially a functional endpoint in some sense of physiology and pathophysiology. For example, starting with DNA, transcription of RNA and then translation into protein, and activated proteins act as enzymes, catalyze various reactions related to signaling, metabolism, and so on. And metabolites, changes in metabolites are actually a reflection of changes in other processes related to genomics. So one of the cool things about metabolites is that typically, as compared to other omics measurements, they can be easily measured from serum or blood. And in many cases, they are also a direct mirror of the environmental influences. For example, if there is malnutrition, whether we exercise or not, and the medicines that we take, those are often reflected in our metabolome. So some of the challenges that we are looking at from the data and metadata perspective in metabolomics are: how can we organize, store, and use metabolomics data? How can we analyze the metabolomics data and interpret the results? How can metabolomics data be integrated with other omics data to provide a systems view of the underlying biology? And finally, what are the implications of metabolomics for systems medicine? So with these, almost 13 years back, NIH started a common fund program, of which Metabolomics Workbench, our National Metabolomics Data Repository, was one of the resources. What I have shown here is a snapshot of the different types of assets we have in Metabolomics Workbench. For example, now we are approaching about 4,000 studies, more than 400,000 samples. And out of the close to 4,000 studies, almost 1,600, more than 1,600 are related to human studies. So as such, this is a really rich resource. And over the years, we have seen that there is a continuous growth in the number of studies that have been deposited into Metabolomics Workbench. And we believe that it's growing strong. So the overall strategy to manage this resource is that we have a user interface through which experimentalists and related teams can submit their metadata and data to Metabolomics Workbench. In case of targeted data, much of the information directly goes into a relational database, which is composed of more than 50 different tables. Then these data and metadata are accessible to a number of tools that we provide on the workbench for analyzing them and visualizing these data sets. In case of untargeted data, where we generally have to deal with a large number of large files, the metadata is put into a relational database, whereas the large files goes into a big data file system for further processing and cleaning, so that it is again available for access and use in the tools that we have. So overall, in terms of the capabilities of the workbench, users can submit their metabolomics data. In fact, nowadays, majority of the journals require one to deposit their metabolomics data. before a paper which includes metabolomics data can be published. One of the key aspects is the harmonization of metabolite names, which I'll talk a little bit more later. The ability to query and search the metadata and data related to studies. Then the ability to download the data for further custom analysis. And for persistence, we use the document object identifier for the project so that even if we were to change something locally for the end user, the resource remains, ID remains persistent. Besides the data and metadata, we also provide a number of tools on the workbench that can be used to visualize and analyze the data. In order to facilitate sharing this data and metadata, we use a common format known as the MWTAF format, which is essentially modeled based on UniProt to make, which is both human readable and machine readable, because eventually we want to make it AI ready. We are not there yet, but we are moving in that direction. So two main sections of MWTAF file format are metadata section, where we have information related to project study, subject and biosample, chromatography, etc. And in the data section, we have the actual information about named metabolites and the actual data matrix. And in the case of untargeted data, the names of the files. Then the users have the ability to provide additional information as comments. And we also have a parser available written in Python to facilitate the conversion between the MW or TSV format and JSON format. There are a number of ways we can search the data and metadata related to studies. For example, we can look at the summary of all the studies. Then we can look at bubble plots with respect to various types of metadata fields. For example, what is shown here is in terms of a sample source, we see that blood is one of the most prominent sample sources that we have seen. And then we also have the ability to perform free text search to find information on the different studies. So there are a number of interfaces for facilitating the search. One is one could search for distinct values in the different fields, such as sample or tissue source, species, then information like institute or year submitted. Besides, one can specify certain keywords in the project and study title or summary field. In addition to that, we provide the option to perform a full text search, which will, with that, one will be able to search for any combination of keywords, including phrases, across all the metadata fields. We can find more detailed information about the individual studies, where we can find the information for the data and metadata. And then we can look at a number of statistical tools as listed here. For example, one for the metabolites related to the nomenclature, one can look at the structure of metabolites and their classes. And then we have a number of tools for univariate and multivariate analysis, such as correlation and clustering, classification, etc. So by providing both the access to the data and metadata and also a number of analysis tools, we are trying to balance between both the infrastructure data aspects as well as the innovation aspect, so that once a user finds, based on some initial analysis, that this is a good study to further follow on, that helps them decide whether how much further resource they should invest in this in terms of downloading the data and then doing further analysis. In order to promote harmonization and interoperability, we use controlled vocabularies, and slowly we are moving towards using ontologies in Metabolomics Workbench. For example, for metabolite names, we use the RefMet nomenclature, which provides a unified way of for the metabolite names. For species, we use NCBI taxonomy IDs. For the diseases, as of now, we use a controlled list, but we are in the process of transitioning to using disease ontology IDs. While we will still allow the users to specify the disease name as a free text, in case they are not able to find a suitable term in disease ontology or phenotype ontology. Similarly, for sample sources, we currently use a controlled list of terms, but we are moving towards using a mix of Uberan ontology for anatomy or cell ontology and cell line ontology as additional options for specifying the sample source. Currently, we are in the process of testing these things through a user interface for the curation team internally. And depending upon how our experience goes with that, we will refine it and then open it for the users as well. The second key aspects of Metabolomics Workbench is the RefMet for metabolite name harmonization. So RefMet is a standard reference nomenclature for the metabolite that we have observed in the metabolomics experiments that are deposited into Metabolomics Workbench. The basic idea is to use the most common name for these metabolites, which facilitates comparative analysis across different metabolomics studies, and also it enables mapping of RefMet names to pathways through integration with the biochemical pathway tools. As of now, we have more than 187 unique metabolite species in the RefMet database. To give an idea about why metabolite name harmonization is actually critical is an example here where the metabolite called lysophosphatidylcholine 1600 is the same metabolite, but across different studies, we have seen that almost 20 different names have been used. So if you want to do any cross comparison across the studies or possibly with other databases, it's really critical that we use a unified nomenclature for these metabolites. In Metabolomics Workbench, we store a lot of raw data deposited by the submitters. Much of that data is LCMS data. And with a total of about 70 terabytes of raw data in NMDR, which continues to grow, we encourage the submitters to upload the raw data in open source formats like MZML, MZ, XML, etc. And we are also encouraging the submitters to submit data on the reference standards and pooled sample data, because over time, as we are also doing some analysis of the data in related projects, we realize that the pooled sample data and reference standard data is really critical for secondary analysis. So it's really important to provide the raw data for two reasons. One is then down the line, we can verify the annotations and assignments that are included in the study results. And also, as in the future, if new standards and validated mass spectra become available, then we can reanalyze this data. In terms of the user's ability to be able to access and download this raw data, during the submission stage, we require the users to provide a table that links the sample names with the raw data files. And then on the actual study details page, we provide the option to look at the details of different raw files without actually having to download the entire archive, because that could be a really large file running into gigabytes and in some cases a terabyte or so. And then one has the ability to look at the actual spectral data contained in those raw files using GNPS dashboard, or in some cases ADAP KDB pipeline. Then we provide a number of tools for both for the users to be able to find information on this data and metadata, and also perform some analysis. And one of those tools is Metastat, which is essentially a query builder using which we can identify what are the studies corresponding to certain disease or species or sample source, etc. For example, in this case, we are looking at all the studies which have LCMS positive ion mode data in humans with blood as the sample source. Now, for sample source, we refer to sample source at a higher level category rather than at a fine-grained level. For example, here blood essentially refers to whether it's a blood, serum, or plasma. So that is something to keep in mind when searching these studies. Then we have a REST interface through which we can extract much of the metadata and data we have in Workbench in different contexts such as compound or study, etc. Besides those tools to access the data and metadata, we are also trying to develop a number of analysis tools, one of them being MetNP, which is essentially a workflow in which, starting with the data and metadata that is available for a study at the Workbench, or in some cases, a user can upload their own data. They can analyze to find out what are the metabolites which are differentially altered between two conditions, and then do further enrichment analysis to do biological interpretation. MetNP tool is available for use on GitHub as a Jupyter notebook. And the other tool we have is called MetGene, where if we know some gene, then starting with one or more genes, we can find what are the related reactions and metabolites, and behind the scenes use the KG APIs to extract that information, and then find out what are the studies in Metabolomics Workbench which have those metabolites measured. So this tool is also available on GitHub and as a separate website. So beyond the resources that we have available within Metabolomics Workbench, recently NIH started a program known as Common Fund Data Ecosystem to facilitate searching of metadata across a number of resources towards facilitating integration of multi-omics data and analysis to derive biological knowledge. What is shown here is essentially on the right-hand side, I have shown a snapshot of the information portal for the Common Fund Data Ecosystem, which is essentially composed of a number of data coordinating center and common fund programs, along with the 5 centers, of which one is the data resource center that is jointly managed by us and the links team at Mount Sinai by Professor Abhimayan. And then we have other 4 centers, Knowledge Center, Integration and Coordination Center, Cloud Workspace Center, and Training Center. So just to provide a glimpse into how we can facilitate searching for data at CFD, on the right-hand side, I have shown if somebody searches, for example, for liver, they will get a number of assets from the different centers. And this searching is made available by using a common metadata model known as cross-cut metadata model. That is our C2M2 that is used by all the centers to share their metadata. And besides the C2M2 metadata, we also accept a number of other metadata forms such as gene sets, knowledge graph, and other assertions. So at this conference, we have a number of other presentations related to CFD. We already had some, and one was going on right now in parallel. And then we have 2 other presentations in this session later on. So to summarize, I hope I provided some glimpse into how the NMDR resource is satisfying a number of fair criteria. For example, for projects, we use DOIs, metabolomics studies are indexed at Omics Discovery Index. We share metadata with the NISCFD, and we also plan to share our metadata with the Metabolomics Hub, a Chan Zuckerberg initiative. And then we make all this data accessible through a number of search interfaces and also through REST APIs. We promote interoperability by using controlled vocabulary terms, ontologies, MW type format, and RefNet standard. We facilitate reuse of the data first by providing a number of tools for the analysis on the website itself. And also we provide the option to download the data for further custom analysis by the users. With this, I would like to acknowledge a number of grants that have funded this work over the number of years. And thank you very much for listening to me. And I would be happy to take any questions. Thank you.
Questions? So while taking questions, we'll set up the next speaker who's online, and we'll introduce her after the question.
Thank you very much for the presentation. I had a question about RefMet. So one of the projects we deliver is the GWAS catalog, and we have a lot of Metabo GWAS, both with known and unknown metabolites. And we are looking for an improved way to represent those so you can consume an ontology. So can you talk me through a little bit about RefMet and how that is structured and how it's updated and what it is mapped to for me to understand if we can use it?
Yeah. So I'll provide some details, and Dr. Oinfei, he is the one who is a lot more knowledgeable. So I think one of the unique aspects of RefMet as compared to any other ones is here we provide the option to use names across various levels of the measurements in this. And what I mean by that is, in some cases, you don't know the precise structure, but you know parts of it. Okay, so in that case we have one way of naming it, right? And then we one can go at even further one level up where we have even little bit lesser information. So we provide I think there are 4 levels of the hierarchy in which we provide the nomenclature in this one. I think we can discuss after later. Yeah.
Thank you.
Yeah.
Any other questions? All right. Thank you very much.